This is the component 2, yes, component 2 in-class questions, up to question 10. When the kidneys are damaged by disease, they might not be able to excrete urea, so this is about homeostasis in the kidney. Here's a dialysis machine, artificial kidney, and you might not know a lot about this, but you don't need to know a lot about this. Everything that you need is in the diagram. A couple of things that we might just make clear from the diagram. We've got some arrows on here. It does say movement of blood, but you can see the arrow is going that way, and the movement of the fluid is going in the opposite direction. See blood moving round and the fluid moving round. Refer to the diagram above. We're definitely going to pick out some points from the diagram above. So 4 marks, so we can possibly pick out 4 things from this diagram. Describe and explain. Describe and explain how the dialysis machine makes filtration, and the key thing here is more efficient. So we're transferring urea from the blood over a membrane into this fluid. Okay. So we think about transfer from one place to another, and what sort of thing makes the transfer or exchange much more efficient. Well, what have we got here? We've got replacement of this fluid. So if we replace the fluid, so why does stuff move? Stuff moves along a concentration gradient. And what is helping here with this concentration gradient? Well, fresh fluid. So dialysis fluid is constantly replaced. Constantly replaced. That is describing the diagram. And then how does that help? That maintains a concentration gradient. Maintains concentration gradient. Then we might remember from fish, right, suspicious of all these arrows going in different directions, so there is a counter current flow. Which also maintains that concentration gradient across the entire length of this tubing. So we've talked about the fluid, we've talked about the directions. Anything else that we can say from there? Well, we have got lots of very small tubes. So that's 2 of our points, and we've got a large number of tubes. So that will increase surface area for exchange. And it says they are 20,000 and its pore size 0.1 micrometer, so it's permeable to these. It's permeable to what we need to get across, but not permeable to other stuff like big proteins and things like that. So pore size is 0.1 micrometers. So selectively permeable. Lets urea through, hopefully, and not kind of our large plasma proteins. So we've referred to the diagram above. All the examples come from the diagram above, and we're going to go things that make it much more efficient. So we're thinking about exchange, and we've described the feature from the diagram and linked an explanation to get our 4 marks. Aquaporin is a type of protein channel that allows movement through membranes. So we always said that water can get through membranes and osmosis, but actually it's these aquaporins that increase the permeability. And it says it is a protein, and it's only 9 amino acids, so quite a small protein. State the minimum number of nucleotides on the DNA template that strand that would code for this. So just a bit of core concepts revision, and you should remember the triplet code. So we've got 3 bases that would code for 1 amino acid. So if we've got 9 amino acids, it's 9 times by 3 is 27 nucleotides. So straightforward if you can remember your protein synthesis. Now we have from where is ADH released into the bloodstream, so just another straight can I remember. So that's a posterior, so behind bit of the pituitary gland. Posterior pituitary gland. So use the diagram above and your own knowledge. So a bit from our notes and relating it to what we've got here. Explain how the release of more ADH causes a smaller volume of concentrated urine. So you know from your knowledge, more DH, it makes the cells of the collecting duct more permeable. So more water gets reabsorbed. So more water gets reabsorbed into the blood. So we just need to use this diagram. So for example, ADH makes the membranes of the collecting duct more permeable to water. That's our own knowledge. So own knowledge, ADH makes the cells of the collecting duct more permeable to water. So that's our own knowledge, and then we just need to explain what's going on here. So let's tell the story. So ADH here arrives and binds to these ADH receptors. So saying what we see on this diagram. So ADH combines with the receptor. Then what happens? Then we've got this vesicle here with these aquaporins. We're going to talk about those. So vesicles fuse with the membrane, and then that's sticking more of these aquaporins into this membrane where they can then be used to let more water through. So now there's more aquaporins in the membrane. So we're just taking that from the diagram. Why would water move? Water would always move down a water potential gradient. So the water potential of the filtrate is higher than the water potential of whatever's over here. So you can say cell, tissue, fluid, blood, plasma. I'll say cell because that's where it's actually showing you moving the first bit, but then you can see water moving into the tissue fluid. And if we're ever going to say water moving, we're going to mention osmosis. So water moves in by osmosis. We'd always say that. So we've used the diagram above, we've used our own knowledge, and we've ended up with a smaller volume of more concentrated urine because there's less water over here on the left side of the diagram, and it's gone back into the blood. Ooh, there's more. Diabetes insipidus. There we are, I can say it. And the other one, diabetes mellitus. And one cause of diabetes is a patient not releasing ADH. How does this cause diabetes insipidus? And that's excessive thirst, production of large volumes of dilute urine. So large volumes of dilute urine. So we can relate this to what we've just been talking about in negative. We can say, so it's just told us how ADH works and then said, oh, we're not going to make ADH, what's the problem? Well, the problem is that this water's not going to, this situation, this diagram is not going to happen, and that's our answer. So we can't forget about what we've just done. So we can say the membrane is not made more permeable. So water stays in the filtrate, and so that would be more water in the urine, and that's what you're saying, large volumes of dilute urine. You know, we could explain that. There's only one mark. You know, we could mention there's no aquaporins. You know, take some more details from this of things that don't happen. Aquaporins don't get put into that membrane. But I think we've said enough for one mark. And then the last part, we're relating this to a bit of genetics. Ooh, lovely. A genetic condition caused by a dominant allele big N. Okay. And this changes the shape of the receptor sites. So we can imagine that if the receptor sites change shape, then ADH won't fit in. So that makes sense. And we've got a heterozygous sufferer. So as soon as we see heterozygous, we go different alleles are present. So we must have big N, little n. And has a partner who is a non-sufferer. And it says caused by big N, so the non-sufferer must have little n's, but also must be recessive, because if they had a big N, the dominant allele present, then they would be a sufferer. So that's our cross there. Draw a suitable genetic diagram. Well, we're going to go back to our Punnett squares. No Punnett jokes this time, sorry.
No, tell him his real name.
Oh yeah, it was
Reginald
Crumbly or
something.
Yeah, Reginald Crumpet Punnett or something. Yeah, that's right. Even worse than strawberry, it turns out. So here we go. We've got alleles here. So the possible alleles that we might have from this partner, well, we could have a big N or a little n. So here we go, big N, little n, there's our two choices. And this partner here, little n, little n. Well, I'm not going to write both of those because there's only one possibility. So there's our little n. We do our normal adding up exercise, big N, little n, and this other one, the possibility is two little n's. So we've followed the instruction. We haven't just leapt straight to the answer, we've drawn a suitable genetic diagram, and the probability of them having a child who would suffer. So this, you link this genotype to a phenotype. This is a sufferer, and this is a non-sufferer. And there's one of each, so the probability is 50%. Or they might accept other ways of expressing it. 1 in 2. We can't say 1 to 1 because that's just the ratio of the two types. So we want to answer the question, probability of being a sufferer, 50% or 1 in 2.